Alright, in this video we're going to be talking about how a nerve, or muscle for that matter, gets up to threshold and gets an action potential and actually sends that action potential down and causes contraction. We're first going to talk about something called EPSPs and IPSPs. EPSP stands for excitatory postsynaptic potential, and IPSP is inhibitory postsynaptic potential. So your nerves, in the last video we talked all about action potentials being generated on an axon, causing the action potential to go down the axon and eventually result in contraction if it goes to the muscle. What we hadn't talked about is where those EPSPs and IPSPs come from. Well, on your nerve, you have a soma, or the cell body of the nerve, and branching off of that you have the axon, which we've talked about, but you also have these dendrites, and the dendrites receive information from tons of other nerves. You can see this nerve here is going to send action potentials down its axon, which then converge onto the dendrite of another neuron, and those will release neurotransmitters. Most of the time, if we're talking excitatory acetylcholine, which will diffuse across the synaptic cleft, bind onto receptors on the neighboring neuron, and cause either positive ions to flow in or negative ions to flow in, to make it either positive or negative. Right? So acetylcholine binding receptors will allow positive ions to flow in and bring the next nerve or next neuron closer to potential. So you have excitatory and inhibitory. The way I think of it is a lot like shoulder angels. So this is one of my favorite movies, Emperor's New Groove. We got this guy Kronk, who is trying to decide what to do, and he has one shoulder angel telling him to do something good and the other one saying let's do something bad. Do it, don't do it, or vice versa, right? That's how your EPSPs and IPSPs work. We start off with this resting membrane potential at a negative 70 millivolts or so. In order to get an action potential generated, we have to get up to negative 55 millivolts. So we have to increase, become less negative. That's what's going on in this gray shaded area. To get up to that, we have to get positive ions to flow in or get rid of negative ions. So we get up to threshold. So how do we actually get these things to come up to threshold and then get the action potential to occur? It has to do with these EPSPs and IPSPs. I'll just show you again. We have our postsynaptic neuron. So this is the one that we're measuring the action potential or the membrane potential in. We are trying to get this postsynaptic neuron up to threshold so we can get an action potential and cause a contraction downstream. But this postsynaptic neuron is going to have insight or information or peer pressure from multiple sources. It'll have an excitatory nerve coming in, and when it releases its neurotransmitter, likely acetylcholine, it will bind on and cause positive ions to flow in and get closer to threshold. In contrast, you could also have an inhibitory potential, which will send out an inhibitory neurotransmitter. A lot of times GABA is one of the more common ones. When it binds to receptors, it actually causes it to become more negative and get further away from potential. The more negative you are, the more negative the membrane potential is, the further from threshold, and the less likely it is going to generate an action potential and actually signal for contraction. So eventually, to get an action potential in a motor neuron, you have to have enough excitatory potentials to raise the membrane potential up to threshold. And as we talked about in the other video, threshold, this negative 55 millivolts, that's the voltage at which voltage-sensitive sodium channels open up and allow stuff to flow. If this is confusing to you, I encourage you to go onto YouTube and just look up excitation contraction or neurotransmission. synaptic transmission, I guess, is what's in this one. There are lots of decent videos out there on YouTube. Now, these EPSPs and IPSPs can influence a postsynaptic neuron in multiple ways. This postsynaptic neuron isn't just getting one signal from this EPSP and one signal from this IPSP. In reality, this postsynaptic neuron is going to have multiple nerves, axons, converging onto its dendrites, signaling either to generate an action potential or not. We have multiple sources of peer pressure here. And there are a couple of ways that this excitatory potential or inhibitory potential can get its way. The excitatory potential, for example, we'll use that. One is called temporal summation. In temporal summation, it increases the firing rate. This excitatory neuron really wants to get the postsynaptic neuron to have an action potential, and so it releases its neurotransmitter more frequently. It stimulates that axon or that dendrite more rapidly. Spatial summation would be you don't have the same neuron recruiting it, but if you're using spatial summation, you can have 3, 4, or 5 of these excitatory neurons converging on and all signaling at the same time. So temporal summation is kind of like a classic episode of The Simpsons where the kids want a pool and they're talking to Homer and they're like, "Can we get a pool, Dad?" He's like, "No." "Can we get a pool, Dad?" "No." "Can we get a pool? Can we get a pool? Can we get a pool?" They keep firing and asking the question over and over and over again, trying to break him down. That's temporal summation. And I can tell you with my kids it works. We haven't gotten a pool, but they get their way a lot by temporal summation. They are relentless. Now, spatial summation would be like, instead of just one kid asking for a pool, it'd be multiple kids asking for a pool. Multiple sources of influence trying to either bring up the membrane potential towards threshold or take it away from threshold. So, what mechanisms does the nervous system use to adjust force? We've talked about getting the neuron up to threshold, but what about force within the muscle? So the nervous system controls how much force a muscle generates, and it uses systems or approaches fairly similar to the EPSPs and IPSPs that we've just talked about. Just as a sneak peek, things you should actually already know, you have motor neurons coming out of the spinal cord that then converge down onto a skeletal muscle fiber, a single muscle cell. Every muscle cell is only innervated by one nerve. It's not going to receive information from multiple sources. So when this motor neuron has an action potential generated, the action potential is going to travel down here and cause a release of acetylcholine from these nerve endings, which will then generate action potentials on the muscle cell and cause contraction. The motor neuron and all the muscle fibers that it innervates is called a motor unit. And motor units can vary in size. They can be very big or very small. The more muscle fibers that a single neuron innervates, the more power or force that motor unit can generate. In contrast, the fewer muscle fibers that a single neuron innervates, the more control it can have. So your eye muscles, every neuron in your eye muscles only innervates a couple of muscle fibers, so you get pretty good control. You can't generate a lot of force with your eye muscles, but you can control very precisely where your eye is going. So how do you adjust how much power is going to be generated by your muscle? So now we've gone from looking just at an action potential in a neuron. Now we're going to be talking about a whole muscle here. Let's say like your quadriceps. Well, the first thing is temporal summation again. In temporal summation, just like we saw in the axon, or in the neuron, sorry, you have multiple signals for contraction. So every time you get an action potential going down this motor neuron, and then it converges onto the muscle cell. It releases a little acetylcholine and eventually results in a small muscle twitch. If you can get those twitches to happen quickly enough, then you will be able to get summation of contractions. So check this out. Here in this preparation we have, here in this preparation we have a muscle that's been taken out of an animal, and the spinal cord is being stimulated by an electrical stimulator, just basically a glorified car battery. And they send a single pulse into the muscle, or into the spinal cord, which then causes an action potential to come down. When you send a single action potential, represented by this little poke up here, or alpha motor neuron spike, it results in a twitch, a very small bit of force. If you can summate those, or add them on together, you can get multiple, you can get the force to add. So if you can get a twitch or an action potential generated quickly enough, they will add on to each other. So that before the first twitch force goes away, you're adding on another twitch. So here we have one action potential causing a twitch. Now we have two action potentials, and before that first twitch goes away, we were able to add on a second twitch and then a third. Here we're having stimulation so quickly that we were able to generate a ton of force. Keep adding on these twitches together. That's temporal summation. Now we have spatial summation as well. Really we'll refer to it as the size principle. When you are trying to generate more force, you can either have a single motor neuron signal more rapidly, which was temporal summation, and get its motor unit to contract more, or you can just recruit more motor units. The more motor units you recruit, the greater the force. And a key principle in exercise physiology is something called the size principle. When you're starting to generate force, you start with small motor units, which are usually type 1 muscle fibers, and then you move on to type 2A and then type 2X motor units. We'll talk more about this in class, but just remember this is a very key principle that you should understand. I'm going to pass that up. Now, how do other nerves influence the activity of motor neurons, or in other words, where do EPSPs and IPSPs come from? One place is from the muscle sensory nerves, afferent nerves. For example, in your muscle, you have these type 3 and type 4 muscle afferents. They sense changes in stretch of the muscle, they sense changes in chemicals in the muscle, and when they're activated, they'll relay that information back to the spinal cord or to the brain and say, dude, the muscle is tired, we've got a lot of acid down here, and they release an IPSP to keep the action potentials from being generated on the motor neuron or in the spinal cord. Those are type 3-4 afferents. And there's some studies I'll show you at another time. Let me skip through all these. I'm running out of time on my free video here. Other things that can influence motor neuron activity are muscle spindles. These can be, they sense stretch of the muscle. If the muscle is getting too stretched, these muscle spindles will relay information back to the spinal cord and cause it to contract or shorten so the muscle isn't getting stretched anymore. This would be an example of an EPSP. On the contrary, you also have other proprioceptors called the Golgi tendon organ, for example, that sense tension in the tendons. When you contract your muscle, you generate tension on that tendon. And if that tension gets too great, the GTO, the Golgi tendon organ, will sense that and actually inhibit the motor neuron from contracting to decrease the tension on the tendon. These are a fail-safe to keep you from rupturing your bicep when you're doing major arm wrestling contests and other things. So go ahead and read through the textbook. Make sure you understand these principles before you come to class. Hopefully this has helped, and I'll see you soon.